The particle size distribution of a material is a function that defines the range of sizes of its particles, powders, or granules by their mass, volume, or other properties. Determining the particle size distribution is essentially practiced in different fields of research, engineering, and quality control. There are various techniques currently used in measuring the particle sizes, and one of these is the electroresistance counting method. The most common one is the electrical sensing zone method. It is used for both organic and inorganic materials in dilute solutions or particles in low concentration whose sizes are in the nano to micro range. Its advantages over other methods include the following. The particle volume analysis accuracy is not affected by optical properties, density, color, or shape of particles. It gives out exceptional resolution. It determines the particle concentration, and it can examine very small samples. The disadvantage is that when the sample doesn't have a low concentration, chances of error increase from what is called a coincident particle passage. This occurs when two or more particles pass through the sensing zone at the same time, and instead of being counted as two, the instrument reads them as one big particle.
Now, we are at the working principle. With this, we start by introducing the setup. The setup consists of a container with an electrolyte with a dilute concentration of suspended non-conductive particles, an inner glass tube with an electrode inside, and an outer glass tube with an orifice. And found outside the outer glass tube is another electrode. All electroresistance counting methods are governed by the Coulter principle. It starts with, upon normal circumstances, charged particles would flow steadily through the orifice. The electrical impedance then increases as non-conductive particles pass through the orifice. In another sense, as non-conductive particles passes through the orifice, it displaces an amount of electrolyte that is equal to its volume. As the electrolytes are pushed away from the orifice, this is consequently detected as a voltage pulse, due to the momentary increase of electrolytes going to the electrodes. These voltage pulses are then related to the volume of the particles and give a number distribution of the equivalent volume sphere diameter of the particles. To understand the concept further, let us consider the theoretical operation given by Micromeritics Instrument Corporation. First, as electrolyte is pumped through the orifice, so are suspended particles. As each particle transits the orifice, it displaces a volume of the electrolyte conductor equal to the particle's own volume, thus reducing electrical conductance across the orifice. An electrical pulse is produced in the electrode circuit, the amplitude and width of which are recorded. Pulse amplitude is proportional to the particle volume. Optional pulse width measurement is related to transit time. The instrument counts the pulses and sorts them by amplitude, thus providing data from which is calculated the particle population by size.
Instrument systems which utilize the Coulter principle are called Coulter counter instruments. The electrical sensing zone technique is also applicable to a wide variety of pharmaceutical applications that include population distribution of cells for tissue cultures and for blood cell counting. Useful applications of electrical sensing zone can be found in the food industry, abrasive industry, environmental monitoring, and many other. Electrical sensing zone technique is also applicable for a variety of sample types like drugs, pigments, abrasives, clay, minerals, construction materials, and coating materials.